Hello, my friends. The time has come that we talk again about some scientific topics. So, therefore, we are back in English. And I want to present you here my first authored research paper in chemistry. And this paper is in fact called a chemically induced CRISPR-Cas13 Cas13 FTPF platform for precise and programmable RNA regulation. So, what is the stuff that we did here? So, to get you involved in the topic, so I hope everybody who sees that has a basic knowledge of what a mRNA is. Basically, the mRNA is the transcript of a DNA, and it encodes the sequence for proteins to be translated by the ribosome. But the mRNA just doesn't pop off from the DNA. It has some modification steps before it's mature. And so the first transcript is the pre-mRNA. And pre-mRNA can be spliced, so basically can be cut at certain positions and then be basically glued together by the so-called spliceosome, and by that means different mRNAs can evolve from pre-mRNAs. So the problem is that during that process of splicing, there can always happen mistakes. And in one disease, the spinal muscle atrophy, there is a problem with the splicing. So normally, where everybody here has the SMN gene. The SMN gene encodes the SMN protein, the survival of motor neuron protein, which is important for the correct function of the spinal cord. So, but some individuals lack this gene, but we have backup genes. Everybody has about 2 to 4 backup genes, which are called SMN2. But those SMN2 genes have the problem that at the splicing position of the pre-mRNA in the around the seventh exon, or the border region of the seventh intron and exon, there's a translational shift which turns the splicing recognized position into a weak one, which leads to the fact that sometimes, or in the most transcript, in 80 to 90%, the pre-mRNA of SMN2 is spliced in the wrong way, which causes the translated protein to be lacking that part, and therefore being non-functional, which causes the symptoms of SMA, the spinal muscle atrophy. So, but for this matter, there is medical or a medicine which is called Elsevier, and the substance is called Ristiplam. What Ristiplam does is that it intercalates into the pre-mRNA, and by that means it leads to a correct assembly of the spliceosome, and thereby leads to the inclusion of the exon 7. So far, so good. Here, this is basically the explanation. So here we see the picture. If we have the pre-mRNA of SMN2, we have this adenosine, which is bulged and doesn't interact in the right way with the U1 subunit of the spliceosome. And by that means the assembly is wrong. Exon 7 doesn't get included. The protein is too short. That is not functional. So Ristiplam basically intercalates here and leads to a correct assembly of the spliceosome, and thereby to a correct uh, translation of the mRNA into a functional protein. So, there is in fact a crystal structure of this SMN2 pre-mRNA U1 complex, which ristiblam lays in here. So, the problem, however, is that this motif here, this GAGU, is present in multiple pre-mRNAs of multiple genes. So, to say FOXM1, APLP2, strNA3, and MED. And the problem is, if this ristiblam can intercalate into the complex here, it can obviously intercalate in the complex here, and thereby not only change the assembly of the spliceosome and thereby the translation of this SMN2 protein, but it can also intercalate into the other pre-mRNA U1 complexes, which leads to the problem that it changes the major pre-RNA and therefore changes the translated proteins, which leads to the side effect of this um, medication. So, which in the easiest case are just something like vomiting or diarrhea, and in the hardest case, very severe side effects. So, the thing is now that we have here the crystal structure or the NMR structure. It's an NMR structure, in fact, in which we see how risdiplam lays in here, which makes it apparent that on the one hand and on the other hand, the risdiplam is basically looking out of this complex, which leads to the possibility that we could modify risdiplam here and here. And by that means, we could basically, our idea was to attach an anchor to this ristiplam and basically direct this ristiplam just into the right position so it can do it actions in the right position but not in the wrong positions. So it just modifies the splicing for the SMN2 but doesn't modify the splicing for the other genes. So the idea, therefore, was that we use our already established FCPF system, which we can explain on this position here. What we already have published is that we have a DCAS9, which is a endonuclease, but catalytically inactive, so that it can still bind onto the nucleic acid, in the case of Cas9 onto a DNA. But if we modify it with this FCPF, we can attach a ligand, in fact, the so-called PFB or perfluorobiphenyl, which does a click reaction with the cysteine. And if we can attach this ristiplam on there, we could make this big chimera by which we have this Cas13, which could then go onto the pre-mRNA because Cas13s, Cas13 proteins bind onto RNA and not DNA. And by attaching our ristiplam, which this click ligand, we could click it onto this and then guide it especially or precisely to the gene that we want. So, what was the idea? That was the idea, and how did we want to perform this? So, this is where my part begins. The idea was basically to redo ristiplam, but you see here we have this position, which is here the upper position. The idea was to attach here the PFB so that we have this clickable ligand to the cysteine. So the idea was basically that I created this ristiplam core, but with an possible exit vector here, so that we could attach this here. So until this point, it worked quite well, but and this here worked also well. So this is the upper part of ristiplam, this is the lower part of ristiplam. However, after this ring closure and the substitution reaction here on and the Bock protection, this cross coupling here didn't work. And by the way, this reaction here is really, really messy because you basically burn this together until you got this black residue, and then you throw on POCl3 and exchange here the hydroxyl group against the chloride, and it's really disgusting. And then you try to activate here via the chloride and then do a cross coupling with this chloride, but that didn't work. So, but as you can see, we have here this other possibility. So I, or me and my boss, choose to try to attach the PFB here. How did we do this? In that case, I basically just did everything until this position here. Or no, this was bought in fact, and I just had to activate it with the Miura relation. Um, and after the successful Miura relation, during this Suzuki cross coupling, I was able to get the cross coupling between this position and this position. And afterwards, it was quite easy going. Under nitrogen protection, I attached this piperazine and cleaved off the Bock group. And after that, we used this perfluor biphenyl. You see, it's a biphenyl with all fluor, so perfluorated, and attach this acid. And this acid could be attached here onto this piperazine after deprotection with a simple H2 reaction, um, which led to our first conjugation of ristiplan with the PFB. Then I was also able to attach this amine here onto the PFB via this not cysteine sulfur substitution reaction, and then afterwards I just attached the acid-based linkers with different length, which I attached via H2 coupling and then deprotected it with TFA. And after that, the same H2 coupling was performed. So we now had some Risiplan derivatives with different linker lengths and the PFB. So you see, we have here basically a very similar structure. We took out this cyclopropane because it was easier to do it like that, and the pharmacological implication that this cyclobutane did to the structure wasn't that important anymore because we had a whole different structure. Um, so after we did this, we first compared it to some protecs in clinical trials via Swiss ADME predictions, which basically just yielded big molecules are bad for oral treatment. So we skipped this bit. If you want to read it, you can read it. But afterwards, we checked how our substances perform. And for that means, you see, we have here, we first evaluated different primers because we, of course, checked if we have our SMN2 pre-mRNA, how can we basically back translated it to DNA so we can quantify how that works. And that way we found our primers. That's not that important. But then we tested how does our do my substances behave, and for that we tested that if we treat this as those cells expressing SMN2, how is the splicing modified by our substances. So if we look here, we have this ristiplan, and ristiplan clearly leads to increase of the full length DNA or a protein because ristiplan intercalates, so on and so forth, what I've explained before. And then we checked what the different linker lengths causes, and you see with PFB1, so the shortest linker, and PFB2, we get increased amounts of the full length. But here with the longer linker length, we get a less amount and more of the shortened exon 7 missing protein. So therefore, we checked furthermore what happens if we add our CRISPR RNA. So the CRISPR RNA is basically the RNA which is here, this here, which binds onto the Cas13 and then guides the whole complex to the gene. So for that, we designed multiple CRISPR RNAs, and I made a mistake. We used reverse transcription qPCR. So of course we're talking here about DNA and not proteins. The proteins weren't translated. It was just DNA, which were reverse transcribed. Just to make that sure. Just to make that sure. So please don't stop the video before I made that sure. So what we made then, we designed those different CRISPR RNAs, which here is the GAGU, and some was before the GAGU, some was on the GAGU, and the last one was after the GAGU. To see basically if we have here our pre-mRNA, when we form the complex of the CRISPR RNA and the Cas13, Cas13, how does it work? Is it better if we direct it directly here, a bit before or a bit behind? So what we checked then is basically we took our different here we took again our ristiplam because we always had this positive control, and so okay, it makes here the full length splicing product. And we checked what happens if we use our two best derivatives, the PFB1 and the PFB2, and then we choose different Cas13 species. And you see, with PFB1, nothing happened at all, but with the and most importantly, you see we differentiated here between the FCPF modified DCAS 13 species and the species without the FCPF. So we compared basically if it made a difference if we just have this DCAS 13 and our substances, or if we have the DCAS 13 with the FCPF and our substances. And as you can see, we only got a change in splicing patterns as soon as we had FCPF and we used ristiplam PFB2, and only with DRFX Cas13D and DRAN Cas13-13B. Those were the only two ones where at 0.5 micromolar we got an increase in the full length splicing product. You see here it's all quantified. So what we then checked is which CRISPR RNA is the best one. And here we see with ristiplam PFB1, again, nothing happened. But if we use ristiplam PFB2, we see a change in splicing patterns with CRISPR RNA1 and CRISPR RNA2. So what we then finally made, here you see it always. Here you see ristiplam, PFB2, and with those two CRISPR RNAs, we get the best results. So what we finally choose to do is we checked what happens if we have DRFX Cas13D with FCPF, and then we have different concentrations of our best hit, the ristiplam PFB2. And what you can see now is that here we have a ristiplam 1 micromolar treatment, and we have here the 0.001 micromolar with our ristiplam PFB2. And if we apply this DRFX Cas13D and a combination of CRISPR RNA 1 and 2, we get really, really good results. And you can see that our EC50 is about 500-fold better than the free ristiplam. So what we in fact made by this is we got basically our DCAS13, guided it or coupled it via a FCPF to our ristiplam derivatives via click chemistry, and with the matching CRISPR RNA, we guided it to a certain position, and at the certain position, the intercalation into the pre-mRNA to correct the splicing was 500 times better than the free ristiplam just floating around. But the best part about is yet to come, because you see, if we target the other genes, which are mistargeted by ristiplam, You see, exactly, for example, with FOXM1, Ristiplam uh, changes the splicing. With STRN3, Ristiplam changes the splicing. With APLP2, Ristiplam changes the splicing. With MAT, Ristiplam changes the splicing. Shouldn't happen. But if we take our compounds at a concentration in which it is effectively, you see 0.001 was good to change the splicing in the case of SMN2. Here we see nothing. Here we see nothing, or at least until just we get to crazy concentrations compared to what is effectively. Here you see the basically no increase compared to the non-treatment. Here you see no bands at all. So basically, to go up to our graphical abstract again, to look at this, what we basically achieved is that we have FDA approved and also in Euro approved drug which can change the splicing of pre mRNA. However, this drug has off target effects because it targets the same motif in other genes as well, and there changes the splicing. What we did now, we developed a system where we get a catalytically inactive Cas13, which can be modified with an FCPF, so the amino acid sequence phenylalanine, cysteine, proline, phenylalanine, which then basically grabs the PFB at the with the cysteine because we have a precise substitution reaction of the cysteine with the PFB motif. Onto this PFB, we have attached this ristiplam, and by that construct, we can force the ristiplam just into the right position so that we have the effect of ristiplam at way lower concentrations without any side effects. And this is basically what we did here. And I hope you can follow it a bit. If not, feel free to read the paper. And if possible, cite it. I would be very thankful and very happy about that. And yes, the biggest problem on this whole thing is that yet CRISPR-Cas systems are very hard to introduce into the system because it's rather big constructs, and also our structures or our molecules themselves are really big. You saw it at the Swiss ADME prediction that they are predicted to be very bad, possible to pass the blood-brain barrier or to be orally administered. But the proof of principle stands. By catalytically inactive Cas13, you can basically guide RNA drugs to the correct position. And if you want, you can here read in detail how I made the substances and how all was prepared. Yeah, I thank you very much for listening and hope you had a good time.